Welcome to Emery Pharma Speaker Series. We have a very special guest here today, our very own CEO of Emery Pharma, Dr. Ron Najafi. He's a serial entrepreneur. He's founded many other companies like CP Lab Safety, which is a laboratory safety product manufacturer, Nova Bay Pharmaceuticals, where he took that company public, and now Emery Pharma. For those of you who don't know, he's the passionate director behind our YouTube channel, our speaker series, and many of the videos that go on our Emery Pharma website. Today he has a very interesting presentation on nitrosamines and their role in pharmaceutical drug development. He's a chemist, entrepreneur, inventor, my personal mentor, and someone I admire a lot. He also actually happens to be my father. So please join us in welcoming him.
Thank you, Anna. So today I want to tell you a little bit about nitrosamines and the role they play in pharmaceutical development. And really there's been a lot of anxiety and questions around nitrosamine, and this has been going on since the late 1970s. And then it died down in the 80s and 90s, and now it's picked up again. So I want to kind of show you a little bit of a chemistry. So don't get scared. This is really high school chemistry. It is really, we're showing an amine here, a nitrosonium ion here, a very powerful electrophile, reacting with an amine, in this case a secondary amine, so you form a nitrosamine, or you call some nomenclature, you can say N-nitrosamine, and this here is your amine, here is a nitroso. So that's how the terminology and nomenclature works. So nitrosonium ion, and NO plus, is a nitrosating agent, and it's also called nitronium ion, and it's often produced, not always, but often produced by the reaction of sodium nitrite and HCl. Having said that, obviously since 2018 there's been a huge, again, uproar of nitrosamine scare around literature. All you have to do is Google it and you'll see. FDA's expectation from the industry is pharmaceutical industry has to be responsible for understanding potential of forming nitrosamine. They need to understand the impurities that are being formed around nitrosamine. They need to know how to manufacture a product. They need to expect potential problem, nitrosamine formation, and so forth and so on. So these are, European medical authority is very similar to this. You can look that up as an expectation. So what is Emery Pharma's role? Since 2018, we've been dealing with nitrosamine initially from the angiotensin II receptors, Valsartan, Losartan, various blood pressure medication. And in fact, when this came up, my mother was taking Valsartan, Losartan, and so it was really personal to me, want to know what's going on. And we were contacted by a number of people, wanted to know what the situation was. So we got involved in doing some nitrosamine analysis using gas chromatography, which is really the go-to instrument for detection of these highly volatile nitrosamines. And of course, we got involved with Zantac, and that's been detailed at length on our website and in the literature and everything. We've also filed a petition with the FDA where we recognize that ranitidine, which is also known as Zantac, if subjected to heat or moisture, can fall into a degradation pathway, which is not very well understood, but we know it produces NDMA. And so Emery Pharma was one of the very first companies that recognized that And on January 2nd, we filed a petition explaining the NDMA formation. And as a result of that, within 90 days, FDA decided to officially withdraw the product off the market. So we know a lot about NDMA, and since then we've actually done a lot of investigation. Some of our team members that I want to acknowledge, Dr. Rakesh Jain, Dr. Neil Bose, Dr. Ryan Chu, they have all been involved in nitrosamine analysis and investigation. So in many ways, this investigation for us is personal. We want to make sure, I know people were taking Zantac for many years. I know a lot of people taking Valsartan. So we want to have safe drugs. So how do we ensure safety of a drug? So to ensure safety of a drug, it's just not, oh, let's synthesize the drug, compound A could go to B, to go to C, various things, and then here is a final product. We put it in the pharmacy shelf. It's more complicated than that. And one really has to do it right and to be able to properly assess pharmaceutical safety. You need to have control on a lot of different things. So your supply chain needs to be controlled. Your supply chain are your components you're using for your drug. Your solvents, are you using recycled solvent? If so, do you have any control around impurities that could be within your recycled solvent? What kind of packaging are you using? Packaging could be a potential source for nitrosating agents. And other types of various products that go into supply chain. Process-related activity this involves synthesis, and you can see the manufacturing. This is looks like a little pilot plant. You're manufacturing, you're synthesizing various compounds. So you need to examine your process. And I've spent years in, as Anna mentioned, at Aldrich Chemical Company, where we would synthesize things in the lab, then we will take it to a pilot plant. So you want to consider what happens in a small scale, large scale. If you're changing a specific reagent, there's an impact. And those impacts need to be looked at in a very intelligent way and with consultation with literature and everything, so to avoid an undesired impurities. And last but not least, at the end, you produce your API, you produce your drug product, you put a pill together. Now, the pill is maybe 1 gram in weight, but only 350 milligram of it is the drug. The rest of it is, the remaining 650 milligram is excipients. We call it in pharmaceutical industry excipients. It could be sugar, it could be cellulose, it could be various other things, binding agent, magnesium sulfate, various things to bind the pill together and also to release the film. So those types of agents also could be a potential for nitrosation forming activity. So you need to consider that, and of course you need to see the interaction of your API with your these excipients. And the way you learn those interactions is through stability study or through force degradation, where you heat it intentionally, you expose it to various conditions to see whether it forms various impurities. And by law, you have to disclose impurities at 0.1%, 0.15%. But NDMA impurities are much lower than that. So you have to follow various guidances as it relates to impurity analysis and as it relates to, for example, various guidances from International Committee for Harmonization. We call it ICH. Like in this case, mutagens are, the guidance for mutagens control are ICH M7, where There is a very detailed guidance what you should do and what are the compounds and how you should treat them, etc., etc. So just to give you a little bit of more chemistry, okay, this is going back to your Chem 1A. So here is your NDMA, N-nitrosodimethylamine, N-nitrosodiethylamine, this is your ethyl. Here's your ethyl isopropyl. Here is isopropyl, isopropyl. So you could have a lot of different things. So you could have probably hundreds and hundreds or maybe thousands of different nitrosating agents. So you need to have access to proper methodology, analytical methodology, such as LC-MS, MS, a high resolution mass spec, and so forth, so that you could actually pick up these nitrosating agents. Now, for example, where's NDMA coming from? For some of us who've been in pharmaceutical or chemical industry, you have heard of DMF, dimethylformamide, one of our favorite solvents. It's a wonderful solvent. We call it polar aprotic solvent, which is it means it's like water, but it doesn't have hydrogen. So it's polar aprotic solvent. The problem with this solvent is when you heat it, it actually, small amount of it, maybe less than a tenth of a percent, maybe hundreds of percent of it, it breaks down from here. So it offers you that dimethyl entity that you're looking for. And there's another complex reaction here that goes on to form a diethyl version. So this becomes this, and this becomes this, and so forth and so on. So you have to have a sense for what happens in the chemistry so that you can actually be able to have some predictive engagement, and also you also have to have the instrumentation to have a reactive engagement. So just what I'm going to be sharing with you in the next hopefully 15, 20, 30 minutes, I don't know exactly, but we're gonna show you the possible sources of nitrosamine. Again, it's simple stuff. Method of formation, reaction with nucleophiles and electrophiles, again, simple organic chemistry, toxicology, toxicity, mutagenicity, carcinogenicity, solvents, reagent, acceptable limit. So NDMA, there is an acceptable limit that you can have according to FDA, European Medical Authority. So you can have some level of NDMA, not too much of it. And analytical methods that has been validated by various companies, including ourselves, FDA method, EPA method, and of course, I'm going to give you some examples of some of the human drugs that have been subjected to the problem, and of course, challenges and our Emery Pharma's contribution. So nitrosamine, as mentioned before, it came, it bubbled to the surface again after late 70s. Again, it bubbled in 2018 through the finding of essentially Novartis, who reported that there was some NDMA in Valsartan they were purchasing from overseas. So again, nitrosamines and the attention to nitrosamine came to being in the 2018. Again, it bubbled to the surface just like in the late 70s, with nitrosamine being in the cold cuts, in food, in the deli section. Now we're having nitrosamine in drugs, Valsartan. And then of course it is a class of compounds that are known to be carcinogen and mutagen and potentially cancer causing, potentially. And evaluation of nitrosamine in drugs has been a big challenge for pharmaceutical industry, and you really need to have the right tools, both from a synthetic chemistry point of view as well as analytical chemistry point of view, to be able to predict and do proper risk assessment. So first of all, this presentation is going to go on our website. You're welcome to take a look at it again, and we have a lot of references on it, so you can take a look. The API and drug products root cause analysis must be performed according to the FDA. You have to if you see NDMA, you need to conduct those. You need to know what are the acceptable limits for NDMA, NDEA, various forms. So NDMA, for example, is 96 nanogram. For NDEA, is lower, it's 26 nanogram. So it really depends on toxicology and literature, what's been reported by FDA, EMA, and so forth. And basically, there are reporting limits that need to be considered. So nitrosamines are really not just limited to pharmaceutical industry and pharmaceutical products. You could be forming nitrosamine when you cure your meat, you're cooking, you're barbecuing. You could have nitrosamine in water. There is actually a limit for nitrosamine per FDA in water. When you do disinfect the water, and you use trichloroamine or dichloroamine, you form nitrosamine. So that's a good place to look at. Detergents, solvents, cosmetics, and of course drugs, tobacco, smoking generates nitrosamine. So here's, again, it's very simple chemistry. I don't want to, I cannot stress this enough. Sodium nitrite is a big source for nitrosamine. If you have sodium nitrite in your food, in your packaging, if you're using sodium nitrite to neutralize sodium azide, for example, which was happening in valsartan, then you're gonna have to have nitrosamine forming. Nitrosating agents are being formed. Literature also reports if you have electron withdrawing group on a nitromethane, that becomes a potential source of nitrite. So the chemistry of nitrosamine is really very straightforward. Sodium nitrite, plus acid forms nitrous acid. So nitrous acid gets protonated again and form this water. This guy leaves, and you form nitrosonium ion. And then nitrosonium ion then does its, sort of dirty chemistry. And that becomes the NDMA or NDEA and various things. And irrespective of whether it's primary amine, secondary amine, or tertiary amine, you form nitrosating agents, okay? I'm not going to get into the chemistry too much. Here in the primary amine, you have a straightforward chemistry that forms this, carbocation, a methylating agent, which gets very quickly hydrolyzed with water. In the secondary amine, however, you end up with nitrosamine, and that stops there. And then your CYP enzyme is the one that actually converts it to cancer-causing, potentially alkylating agent. So here's how tertiary amine has been speculated and has been studied by a number of people. Look, just for your information, look at the dates of these publications: 1972, 1978. This is when I started going to college, back in the day. But this is actually when things were really heating up with nitrosamine back in the day. So you're forming nitrosamine easily there. And then, of course, there is N-protonated nitrosamine also undergoes denitrosation with various nucleophiles. So in other words, if you protonate under an acidic condition, nitrosamine actually converts to other things. So that's what we've actually observed, where nitrosamine is actually not that stable. It actually transfers its nitrosating agents onto other things. So it's a very labile sort of a moiety. And again, more references here for you. And there is also O-protonated nitrosamine that also has the potential of forming. So lots of very good publication by a group out of MIT, a recent publication 2021. It's worth looking at. It's good exercise for your chemistry class. And then of course, what happens in your nitrosamine, your NDMA chemistry, where NDMA in the body goes through the your liver and and then gets in contact with CYP enzyme, and the CYP enzyme does a job on it, and this transformation, and forms this reactive species, which is the methyl diazonium ion, which is really a bad guy, and it becomes a source of an alkylating agent. And then that reacts with the DNA, specifically with guanosine and guanine, and effectively it intercalates into the DNA and potentially causes mutation there. So basically, we talked about DMF and triethylamine and various things. So here are just some of the chemistry that happens with DMF. Hydrolysis of DMF, forming dimethylamine and then getting nitrosated, or how triethylamine gets converted into N-nitrosodiethylamine. These are some of the intake levels, acceptable intake levels. So the companies, pharmaceutical industry, need to be familiar. If they see nitrosamine, they see NDMA, NDEA, they need to know that these are the limits that they need to put control around their API or around their drug product. Some of these analytical methodologies were developed by Emery Pharma and of course European Medical Authority, FDA, have also developed and used these analytical techniques. A lot of liquid chromatography, mass spectrometry, high resolution mass spec, and gas chromatography are used to determine NDMA. And NDMA, in the years prior to mass spec in the 70s, how did they measure NDMA? They used gas chromatography. A flame ionization detector, various, and the flame ionization detection is extremely sensitive, and they were able to measure it. Lots of literature for you if you want to look at some of these literature, and of course FDA has good guidances, and so does EMA. I want to just share with you a little bit of a history with Valsartan. In 2018, that's when NDMA bubbled up through Valsartan here, and everybody's attention was focused on various drugs, and then of course there's losartan, and then rifampin, and ranitidine, and various things came into the picture. And it continues. The more you start looking, the more you realize that nitration happens in a lot of different things. So you have to have control around it. So we're going to start with the Zantac. I'm going to give you a very quick sort of summary of what's going on with Zantac. Here, or ranitidine, here is the molecule of ranitidine. You see you have a dimethylamine, and you have a vinyl nitro compound, and the chemistry and mechanism of action of how this molecule converts into ranitidine or converts into NDMA is not straightforward. There are lots of publications. Some of them are here, and also recently I saw a publication by Paul Harmon. Also I suggest looking at that. He has good sort of mechanistic proposals around it. But what we know is that, of course, Zantac was on the market for 37 plus years. It is an H2 blocker used for GI issues, used for various gastroesophageal reflux, stomach ulcer, various things. NDMA was found in ranitidine by Valsartan and later by us and our citizen petition. And then, of course, investigation ensued, and a lot of work was done by GlaxoSmithKline themselves around the root cause analysis. European Union did some investigation. They found that a 150 milligram pill could contain 1100 nanogram of NDMA. The legal limit is 96. So, here is 1100 in a 150 milligram pill, and you can have up to 300 milligrams. So you could be ending up with 2000 nanogram or so of NDMA. Australia reported 2100, Korea 8000, and of course through our study we found that heat and moisture play an important role in this conversion. Some proposed mechanism of NDMA formation from ranitidine is here. Again, these are some little bit of a hand waving by experts around how NDMA can be formed, how nitrosation can be formed here. And some of that speculation was done by GlaxoSmithKline themselves. And so Glaxo's GSK's conclusion was NDMA was not detected in freshly batches of drug substance, suggesting not formed during manufacturing. So that means it was not an impurity. NDMA was generated in drug product or finished drug product due to inter or intramolecular degradation. So it means that the molecule, two ends, either reacted with themselves or two molecules reacted from one end to the other end. The rate of NDMA formation was elevated in stability study due to increase in temperature and relative humidity. And they also reported crystal morphology plays a role in NDMA formation as well. So what about Valsartan? Valsartan is an entirely different story. Valsartan molecule, you have this tetrazole moiety. And this tetrazole moiety is formed, again, Valsartan, we can talk about what it's being used for. We talked about it earlier. It's a blood pressure medication and so forth and so on. Initially invented by Novartis, sold under the name Diovan, and was marketed by Novartis and Sandoz. But the tetrazole moiety is really where the problem occurs. And I'm going to take you through that a little bit. The highest level reported was 19,000 nanogram for 320 milligram. So you can imagine if somebody is taking, these are chronic medication, these drugs. We call it, in pharmaceutical industry, we call it lifestyle drugs. So lifestyle drug means you're taking this and you're enjoying your good food and everything and manages your blood pressure. So if you're taking this for 20 years, you have a high exposure to NDMA. So in terms of chemistry, just to really make it very simple, Novartis, the inventor of the drug, was using tributyl tin azide in this cyclization. So you have nitrile going to tetrazole. This is a classical chemistry where 3 nitrogen is added to one other nitrogen, and you form this basically this is the 3 nitrogen, this is the nitrogen from the nitrile. So you form this tetrazole. Novartis was using tributyl tin azide, and it was getting rid of the excess tributyl tin without, in a very gentle manner. When this product became generic, the generic manufacturers overseas wanted to actually optimize their yield so they could have a better process, make more money, etc., etc. And they changed the process. Instead of using tributyl tin azide, they used sodium azide. Sodium azide is a potential explosive. So you have to get rid of excess sodium azide. The way you get rid of the excess sodium azide is by sodium nitrite. So when they use sodium nitrite, of course they form nitrous acid, and nitrous acid ends up leading to NDMA or nitrosation. So that was a classical sort of mechanism for valsartan and formation of not only NDMA, NDEA, and all sorts of things. And there were also issues potentially with solvents being recycled and contaminated solvents and so forth. Metformin, very, very useful drug for diabetes. It's the first line treatment for diabetes. And it's an extremely safe drug, good drug. Again, in 2018, 2019 timeframe, they discovered NDMA in valsartan or in metformin. And again, metformin has your dimethylamine, and there is a speculation that some of the packaging of the valsartan might contribute to the nitrosation. So this dimethylamine could fall off, could hydrolyze, just like the mechanism of DMF, where I showed dimethylformamide. And then perhaps nitrosation was found in packaging or in water. Oxidation in water could have contributed to the NDMA. And of course, Januvia is another anti-diabetic drug, very popular and useful. And it was reported that the nitrosation was found on these two moieties from their final product, final drug, and and that was something that the company went to gear to do risk analysis and figure out what's going on. Rifampicin is an antibiotic. It's used for tuberculosis, and it's one of the last resort drugs for tuberculosis, and it's used for drug-drug interaction. It is also getting nitrosated through a mechanism that, again, it's being proposed that this dinitrogen species, this hydrazine species, falls off and ends up getting nitrosated. So you end up with this N-methylpiperazine, and then this N-methylpiperazine gets nitrosated. So in summary, we're very close to the end, okay? Good news. In summary, harmful level of nitrosamines have been observed in many drugs in the last few years. And it could be due to the solvent, it could be due to various reagents, it could be due to the molecular structure of the drug itself. And so we would have to be looking at drug product, drug substance, stability, all of the things that we talked about, excipients, to make sure those are all being controlled and being managed by various risk analysis methodologies. In conclusion, FDA really is requiring pharmaceutical industry to do proper risk analysis for nitrosamine and try to identify nitrosamines in the drug product, drug substance. Some of the things we do at Emery Pharma is we evaluate the supply chain, we evaluate the chemistry, we evaluate basically the stability of the drug with the excipients. We look at API, we look at drug product, we look at the root potential of what is being formed, NDMA, NDEA. Is the drug getting nitrosated itself? Are there some of the byproducts getting nitrosated? Once we see nitrosation, then we determine some acceptable intake levels, and then we try to put controls around those. We try to effectively put a statement, nitrosamine statement together as a result of our investigation that would allow a company to move forward with their drug substance, drug product. And so these are much of the same thing that we discussed, and root cause analysis need to be conducted once those impurities are found. And the challenge with nitrosamine is, root cause analysis are very diverse. You need to be well versed in the chemistry. Nitrosamine chemistry is also complex. Not only you have volatile substances, you could also have nitration on other parts of the molecule. You have analytical methods that need to be super sensitive. And then, of course, mechanism of action need to be looked at. And, of course, Ames test not always is a good way to assess carcinogenicity, and it's really not the last thing that you should be relying upon. We covered this slide at the very beginning. Again, we've been involved with almost most of these drugs that I talked about today. We've done the petition for for ranitidine. We understand that chemistry, and we're here to help if there's an interest by anybody. If you're watching this presentation and you think you could benefit from a nitrosamine risk analysis by Emery Pharma, please do not hesitate to reach out to us. And thank you.